Case Western Reserve University's Institute for the Science of Origins proudly presents the Origin Science Scholars Program. The Institute advances the scientific understanding and application of origins and evolution of human and natural systems. The Origin Science Scholars Lectures are presented with the assistance of Case Western Reserve University's Siegel Lifelong Learning Program, College of Arts and Sciences, and Media Vision.
Tonight it's my pleasure to welcome Dr. Serpil Erzurum, Chair of the Department of Pathobiology and Alfred Lerner Memorial Chair in Innovative Biomedical Research at the Cleveland Clinic Foundation. Dr. Erzurum is Director of the Cleveland Clinic's Research Unit, and tonight she will be telling us about CoQ and milkshakes manipulating mitochondria to treat lung disease. Dr. Erzurum.
Thank you. Thank you for that kind introduction, and thank you for inviting me to speak tonight. I am going to tell you a bit about lungs, respiration by the lungs, and cellular respiration in the cell and how they're linked. And we'll end up with a lung disease that is related to abnormalities of respiration. So we'll get started. And I'll refresh everybody's memory about the function of the lungs and the heart. And you can see on this slide the lungs, the pulmonary circulation, the heart, and here's the body. So blood returns deoxygenated from the body to the right side of the heart and then is pumped into the lungs where the alveolar sacs allow the blood to come into contact.
Could you use the microphone?
Oh, yes, of course. To come into contact, yes, good, with air, and oxygen is taken up by the blood, so the blood is oxygenated, returning to the left heart, which can pump it to the rest of the body. All that oxygen is primarily used by one small organelle found in the cells of the body, and I know you've heard a lot about this in the last two lectures, and that is the mitochondria, which produces the energy for the body and during that process uses oxygen. This is a better view of how the air passages and the blood flow are matched. The lung has air going along with blood in all its branches until it gets down to the very terminal spaces where the capillaries form and perfuse amongst the air spaces called the alveoli before the oxygenated blood comes back through the pulmonary vein. I really enjoy looking at the lung under the microscope. It shows you that the lung is mostly air and blood and very little lung tissue. And this shows you how thin the walls of those alveolar sacs are. The center is clear. It's air coming in from those air passages as you breathe in. And here we see the small blood vessels that percolate amongst the air spaces. And you can see one red blood cell passing along the vessel and then returning oxygenated to the left heart. This is a higher magnification, again showing that alveolus or gas space in the lung. And here you see the outline of a very clear red cell, and see how thin the walls of this vessel is. And then there is also a very thin epithelium lining. That allows very close contact of this red cell and the airspace. One of the most beautiful pictures of the lung is if we perfuse it with matrix that solidifies through the blood vessels. And here, this is showing you the lung tissue stripped away after we've embedded the lung in matrix that fills all the blood vessels. And you can see how the blood vessels just can outline those gas spaces. Look at how dense this capillary network is compared to a skeletal muscle distal in your arm, for instance, where you have arterioles that you're probably used to thinking about, a single vessel, and a capillary between them where the gas oxygen can go out and CO2 can come in, and then returning by a venule to the right heart. So the two circulatory systems exist, but perform very different functions: taking up oxygen, letting CO2 go, releasing oxygen, picking up CO2. And that's the cycle of the respiratory system, and it does include the heart merely as a pump. This is a human lung that is breathing, and we've taken an image of that individual as they breathe, and we've put vectors to mark the flow of air down the tubules that are those air passages. And what you can see is that the air moves in and out, and the greatest air movement is toward the bottom of the lungs here, and this is the top of the lungs. And that is because most of the blood flow is to the bottom of the lungs, and that way you get most of the blood flow and most of the air going to the same place, so the function can be optimized for gas transfer, oxygen transfer. That's called ventilation, coupling to perfusion, and it's been known for a long time. Well, there's another very important factor in transporting oxygen and CO2, and that is the red blood cell. The red blood cell makes up most of the blood system. It's produced in the bone marrow, and when it's produced, it is able to pick up oxygen and CO2, and its job is to carry those two gases to the distal parts of the body, and then, of course, into the lungs. How does it do that? The red blood cell has a molecule in it, and it is packed full of this molecule called hemoglobin. Hemoglobin is made up of 4 chains or proteins. And within each of those 4 protein molecules that come together to form this entire molecule, there is a special structure called a heme molecule. It's shown here as a much larger figure, and it's inserted into each of these chains. This heme molecule is produced in your cells, and an iron is inserted to the center of the ring. That iron then is linked to the protein through this bottom position, and it leaves the top portion of the iron molecule free to bind oxygen. It can bind 4 oxygens per hemoglobin, and each red cell has thousands of these molecules. So they really are a transport vehicle to carry oxygen load. Once it gets to the tissue, the oxygen is released. The system that allows for uptake of oxygen in the lungs and release in the tissues is really interesting, and I'd like to explain that to you. This is a graph that shows the oxygen tension, oxygen concentration, and it's going from 0 to 100. In the arterial system, or the lung, the oxygen availability is around 100. So in those gas spaces, very close to the capillaries, the oxygen tension is high. In the tissues, oxygen tension is low. It varies between 20 to 40. The tissue oxygen tension determines how much oxygen is on this molecule. In the lungs, at the higher tension, this molecule is loaded completely or saturated 100%. Every single hemoglobin molecule in every one of your red blood cells is carrying oxygen in the lung. And as it's loaded and moves out into the arteries, pumped by the heart, it traverses through those small vessels out to the tissues and the capillaries. As it gets out into the tissues, you can see that the oxygen tension in the tissues as you're moving or talking or breathing drops. And as that tension drops, this molecule changes conformation and releases oxygen. The saturation falls, and it falls very quickly. You want most of the oxygen to come off when you're in the tissues. And so this rapid decline is something that is a cooperativity of these four chains that allows for this release. And then the deoxygenated, low saturated hemoglobin returns to the right heart to pick up more oxygen and repeat the cycle. And that is the physiologic respiratory cycle, and it involves the heart, the lungs, and red blood cells. I am showing another molecule here. This molecule is called myoglobin. Myoglobin only has one protein chain as opposed to hemoglobin that has four. Myoglobin is found in tissues at very high levels in skeletal muscle, for instance, and heart. Myoglobin is different in its oxygen uptake. It also has that heme molecule, and that heme molecule has an iron that's capable of binding oxygen. Because myoglobin is found in the tissues, it lives in an oxygen tension of between 20, 40, and 60. You can see that as the red blood cell hemoglobin releases oxygen, myoglobin in the tissue catches it. Oxygen comes off the red cell, hemoglobin, and moves to the myoglobin in the tissue. Myoglobin is the molecule that holds on to oxygen for you in your tissues. This is particularly important in your heart that always has to keep beating. And in skeletal muscles, as you run or jump or move quickly, you have to have a steady availability of oxygen in the tissues to hold on to oxygen for use, and we'll see by the mitochondria. There are many ways to accomplish the respiratory cycle. And I think you can recognize this as a dolphin. It is a mammal and has a blowhole. And this dolphin has a very large lung, larger than ours. But he can dive very deep beneath the ocean and stay down there holding his breath for upwards of 45 minutes to an hour. He has the same red blood cells we have. He has the same skeletal muscle we have. What's different for the dolphin? Well, he gets up to the surface and he breathes and he breathes and he breathes, and he loads as much red cell capacity for oxygen, and he dives, and the oxygen comes off and off and off, and his saturation falls to 0. But he doesn't feel short of breath because the dolphin, like most diving creatures, have adapted a way to keep more oxygen in the tissue, and that is that his muscles have 10 to 20-fold more myoglobin than we do, which means he can be very happy swimming around with plenty of oxygen in the tissue, even though there's no oxygen on his red cells and he's not breathing. So, in fact, the more myoglobin even humans have in their skeletal muscles, the longer you can hold your breath too. It's directly related. So I wanted to explain that to you because that becomes very important as we talk about the next part of this presentation. And we start to talk about how cells use all that oxygen. And I can take questions now if you'd like on this first part.
When a red blood cell that's fully loaded with oxygen on its hemoglobin moves into the tissue for the myoglobin, is there any kind of chemical trigger or something that causes the oxygen to separate, or is it just a diffusion gradient?
This is the question that still is a mystery to us. So I had said that that hemoglobin molecule has 4 proteins. They're not the same proteins. There are 2 alpha chains and 2 beta chains. And when there are the 4 molecules loaded with oxygen, it seems when you load one molecule, the next one is easier, the third one is easier, and the fourth just flips on. And just as well, when you take one oxygen off, the next one comes off easier, and the next even faster, and the fourth just flips off. So there's cooperativity between the proteins by their structure, and when one molecule of oxygen comes off, the other protein changes structure, releasing the oxygen, and then that makes this protein change to release oxygen. I'm telling you that because that's what I was taught in medical school. But there are new theories now that in fact, perhaps those molecules are always flipping back and forth, and oxygen is being held there perhaps by other mechanisms. And I will not talk about that tonight, but it would be a good future talk at this session, yeah. There's a whole different conversation with that.
So we'll talk about the illegal stuff. When the bike racers, Armstrong and so on, they would take blood from themselves, yes, store it, and then jam it, and that's the purpose then, to get more hemoglobins?
Yes, more oxygen carrying capacity to get the oxygen to his tissues. When somebody like Lance Armstrong or an extreme athlete is pushing themselves to performance, even if they're doping or doing something, it is still mind over body. And they are pushing themselves to the very limit of human potential. And the more he has to provide oxygen, the less likely his tissues are to enter into an acidic state. I actually used to measure, I trained in Colorado, and a lot of the athletes would come up to Boulder to train and then go up high and come back. And I remember doing exercise performance studies on some of these athletes that were amazing, where a normal person stops because your tissues are acid and your muscles are screaming in pain. They keep going and going and going. It's amazing. And it is truly, they just want to keep going. And so the little bit extra they can get with red cells or myoglobin or anything else they can do is going to help them go that little extra, and that's what they're driving for.
And they train at Broadmoor Hotel, if I recall it, in Colorado Springs and at the academy.
That's right. That's right. Yeah. Are you an athlete? Yeah. I'm not either. Not anymore.
Thank you for joining us. You've been watching Dr. Serpil Erzurum discussing how the heart and lungs work together to deliver oxygen to the body. For more information on the Origins Science Scholars Program, please visit the Institute's website at origins.case.edu. In the next part of the talk, Dr. Erzurum will discuss mitochondria and their connection to lung disease. Now, back to the talk.
So that was all big organs and things moving around the body. Everybody feels comfortable with that. That's easy. We're going to get into the tissues now, and the tissues are the cells. The cell structure is shown schematically here, and it has a cell membrane that encloses a number of organelles, and the nucleus is in the center. The nucleus has all the genes that make up most of the proteins in the cell and direct the cell to be what it's supposed to be and function in what it's supposed to function in. But everything this cell does would be for naught if it wasn't for this structure. That's the mitochondria. And that's because the mitochondria, and there are numerous mitochondria in each cell, provides the energy for every single function the cell is going to do. And so the mitochondria is the energy engine that drives cellular function and truly life. So let's see how it works. Well, first of all, it wasn't always known about. And I'm showing you here some very wonderful scientists, Dr. Palade, actually, and Duvet and Claude. And Dr. Palade actually directed the Rockefeller Institute in New York for some time. And these three gentlemen identified these organelles under very high magnification in cells, and they called them mitochondria. And they hypothesized, based upon the structure, that it might be involved in energy production, and that's just mind-boggling. This is the original paper. That they could intuit what it might be doing based upon where it was found in the cell and how it looked. I think it's hard to look at it in a flat surface like this. This is a little bit easier. It's a three-dimensional model of a mitochondria. You can see it looks a little bit like a bacterium, from which many people believe it is derived, a bacterium that infected a host cell at one point, and it became symbiotic. It provides energy for this cell to do its thing. And you can see it has an outer membrane, and then there's this inner membrane that invaginates. And then there's a central portion. And this is the double membrane part that makes this organelle very special and lets it do its job. I'll show you how that works. So this is my version of that mitochondria. And you can see this is that outer membrane. Here is that inner membrane. And in between is the space you saw in that 3D picture. It's called the mitochondrial inner membrane space. That is holding in place a lot of different proteins within the central structure of the mitochondria. The mitochondria has its own genome and can make many of its own proteins, but many of the other proteins come from that nucleus in the cell. So the nucleus provides some proteins to the mitochondria so it can survive. It's truly a symbiotic relationship. The mitochondria cannot live alone, and the cell cannot live without the mitochondria. And how does it give us energy? Well, we eat to get energy. Any child knows you eat food when you get hungry. I need energy. And that energy often comes in the form of lipids, carbohydrates, and proteins. If you have a sugary drink and you ingest glucose, that 6-carbon molecule will be progressively degraded. And during that process, energy is released as you break carbon bonds off of it, and you make a little bit of energy as ATP. That's the energy currency in the cell. It ends up as pyruvate. Pyruvate is a 3-carbon molecule, and it enters the mitochondria. There's a special transporter for it. And as it enters the mitochondria, it's converted to another compound that enters the tricarboxylic acid cycle, or Krebs cycle. The Krebs cycle continues to break off carbon molecules, and they're released as CO2. The mitochondria is the source of carbon dioxide that you exhale. As it's breaking down this glucose molecule, pyruvate, and carbons are coming off, there are little molecules that pick up hydrogen. And depending upon what part of the Krebs cycle it is, some of those hydrogen ions go to a protein molecule called complex 1, others go to complex 2. And as they go to those molecules, the compound actually puts the proton in the inner mitochondrial space and takes the negative charge from that hydrogen and passes it off to coenzyme Q. Complex 2 does the same thing. Coenzyme Q takes all those electrons, passes it on to number 3. It's a little chain. Complex 3 passes the electrons on to complex 4 and pumps another hydrogen ion. Complex 4 has nobody to give the electrons to, so he gives it to oxygen. And that's why you need all that oxygen. The mitochondria uses 95% plus of the oxygen you take up from your lungs and gives rise to all the CO2. This is cellular or mitochondrial respiration. Now what happens to all those hydrogen ions? Now you've got a big charge here, positive, negative on the inside, pluses and minuses. It's a battery. It's a battery with stored energy. And it can release that energy like any battery can by short circuiting, and as it allows the plus to come back into the cell, it catches that potential energy and phosphorylates and makes another energy ATP. Except you get over 30 ATPs for every glucose as opposed to 2 just by glycolysis. So if you're going to eat and you want to get every piece of energy out of that glucose molecule, you go into the mitochondria and you have cellular respiration. I want to show you that the mitochondria, depending upon how active your tissues are increased in number or decreased. This is a cardiomyocyte, a heart muscle. Your heart muscle from the day you're born, conceived, to the day you die has to keep beating, just moving every second, minute of the day. No rest for it. It needs a continuous supply of energy. For every one muscle fiber to beat, you have a mitochondria of its own, an energy supply right next to every little tiny muscle. And the heart takes up a lot of oxygen to make the ATP it needs to keep pumping. All right. So the heart requires energy. Your skeletal muscle requires energy. Now I'm going to tell you about a disease of the lungs that leads to heart failure. I don't know if you have read or heard about pulmonary high blood pressure. It is different than systemic blood pressure. So most people, when they say, I have high blood pressure, they're talking about the pressure measured in their arm. Patients who have high blood pressure in the lungs have high pressures in that pulmonary artery that provides the hemoglobin to the lungs for oxygen uptake. And that high blood pressure in the lungs can be caused by genetic mutations or lung diseases or pulmonary blood clots or heart diseases. And they're all linked together in large measure by one thing. They're diseases that are related to low oxygen or hypoxia. It's a big risk factor for pulmonary hypertension. So what happens in pulmonary hypertension? This is that pulmonary artery I showed you at the beginning, and I'm showing you the long tube that as it goes out into the lung, remember I told you it became smaller and smaller and smaller until it's a capillary. There's a lining of endothelial cells, but there's smooth muscle that wraps around this artery like a slinky. And in pulmonary hypertension, there is a proliferation of that smooth muscle that chokes off the distal vessels. So remember that beautiful picture I showed of how the blood is found throughout the alveolar spaces. This is a very small image of that. When you have normal vessel size. In a patient with pulmonary hypertension, their smooth muscle and endothelium proliferate, so you have a pinhole opening. This thing, in a real patient, looks like that. It's hard to get blood through that. And the pressures go high as the right heart tries to pump against that obstruction. You lose blood vessels, they drop out. There's less capacity to take up oxygen in the lungs. The right heart fails, and patients die. I will show you a picture of a human heart who has right heart failure. This is the right ventricle, this is the left ventricle. This is the valve where the blood is coming in. This right ventricle is enlarged 5-fold, and it's even impinging on the left ventricle. The left ventricle cannot get blood out to the rest of the body because the right ventricle is so large it's not very good at pumping into the lungs either. So I work at the Cleveland Clinic, and we have a large number of transplants of people with hearts and lungs like this, and we take them out and put in new ones, transplantation. About 20 years ago, I started to catch those lungs and hearts and study them. And I found the cells were proliferative and showed that they were more proliferative than normal cells. They look the same in the culture plate, but they're faster growing. Now, about in 1920, Dr. Otto Warburg was describing fast-growing cells, and even then he knew about cells using glucose. And he said that when cells started to grow really, really fast, they tended to use more glucose and they didn't take up oxygen. He was measuring it. So I thought, were my fast-growing cells not using enough oxygen, just like Dr. Warburg's? And I looked at control and pulmonary hypertension cells, and at any stage of oxygen consumption I looked at, pulmonary hypertension was nearly half what healthy cells were. Half, and half, and half. They used less than half the oxygen a normal cell should. How were they making energy? Well, all they have left is glycolysis. And that would mean they'd need a lot more sugar. And I measured how much sugar they were eating. And we found that pulmonary hypertension cells ate 2 to 3-fold more sugar than normal cells. We were able to measure that using a test that's derived from Dr. Warburg's Nobel Prize winning work. We can label a glucose molecule with a radioactive compound called fluoro-F18. You label glucose and it's fluorodeoxyglucose. We inject it into your bloodstream, and any cells that are using a lot of glucose take it up very quickly and hold on to it. And then with a PET scanner, we can see where that uptake is occurring. And we showed that pulmonary hypertension lungs were taking up quite a bit more glucose, or FDG, than controls. And more importantly, when we looked at their hearts, we found something extremely surprising. Here is a healthy individual. You can see when I make it black and white, the heart doesn't pick up any of that fluorodeoxyglucose. But you can see the patient's heart does. Not only is the lung tissue taking up more glucose, the heart tissue is taking up more glucose in this disease, characterizing it as a disease where the cell mitochondria doesn't work so well, doesn't take up much oxygen, and therefore they switched to using primarily glucose through glycolysis. And I can ask if you have any questions now that I've introduced this disease.
Say a little bit about the precipitating causes of the hypertension and pulmonary hypertension.
I went over that pretty quickly. So we're actively studying what is the cause of pulmonary hypertension. And it's a lethal disease. It used to be that the average survival was 2 to 3 years once you were diagnosed. We have some medications now that work to open up those blood vessels a bit. The risk factors really are low oxygen. So if you live at altitude, at high altitude, altitude sickness, one of the characteristics is pulmonary hypertension. You come down to normal levels of oxygen and it usually goes away. But chronic mountain sickness is pulmonary hypertension. If you have sleep apnea, your oxygen goes low when you're not breathing at night, that's a risk factor for pulmonary hypertension. Lung diseases, where you tend to have a lower oxygen availability because your lungs aren't working well, that's a risk factor for pulmonary hypertension. But there are causes, or I should say types of pulmonary hypertension where none of those things are present and the patient still develops pulmonary hypertension, and that's an area actively being investigated. We think it's linked to this abnormality in the mitochondria.
Could you discuss any factors that impair the TCA cycle and electron transport?
So the question was, Why doesn't the mitochondria work so well? Is it something wrong in the Krebs cycle? Is the carbon not able to be oxidized as well? Is it something in electron transport chain where the complex 1 and 2 can't transfer it to coenzyme Q? Maybe the oxygen acceptor isn't working for electrons to flow out. Where's the backup? There's been some of my own work and colleagues in Toronto that point to abnormalities in electron transport. I will tell you that pulmonary hypertension is a major problem in the poultry industry. Anybody here know about the poultry industry? It's really interesting. Pulmonary hypertension is a major cause of loss of chickens if you're a chicken farmer, just fall over dead. And a lot of money was spent by the poultry industry, and they found it was linked to abnormalities of complex 2. It's an inborn error. And so, at least in that spontaneous model where pulmonary hypertension develops, it's related to electron transport chain function.
We hope you've been enjoying the Origin Science Scholars Program with Dr. Serpil Erzurum. Dr. Erzurum holds the Alfred Lerner Memorial Chair in Innovative Biomedical Research at the Cleveland Clinic Foundation. In the second part of our talk, we learned how mitochondrial malfunction can lead to heart disease. In our final segment, Dr. Erzurum will discuss Coenzyme Q, which facilitates electron transport in mitochondria. Now, back to our talk.
So what am I going to tell you now? I'm going to tell you about the next step in our studies in this pulmonary hypertension disease. So the questions were great. I did think there was something wrong with the mitochondria. Everything was pointing towards it. They didn't seem to be working very well. So I wondered if there was another way I could measure mitochondrial function. Very hard to do these studies with just these explanted tissues. And I wanted a real way to measure it in people. And I thought, what else does the mitochondria do? It actually does more than just make energy. It's very important for producing that heme ring. That heme that gets inserted into hemoglobin and myoglobin that has the iron in it, that iron is inserted into that ring in the center of this mitochondria. It all fits together so well. And that heme production, the final steps here in the mitochondria, and then it's exported out and inserted into the right protein like myoglobin or hemoglobin. And if you're a red cell, you get produced and go out into the blood and you start carrying oxygen around. If you're myoglobin, you stay in the tissue and you're catching oxygen. So I wondered, was there a way to tell if there was something wrong with the mitochondrial heme production? Well, there's an easy way to tell. In fact, Flint, Michigan, how did they know that those children were poisoned by lead? Lead poisons you by interfering with mitochondrial heme synthesis. It blocks the insertion of iron. Now, if you're iron deficient, you also don't have iron to insert. And maybe if you have pulmonary hypertension, it's not getting in there because the mitochondria is not doing it. If the body doesn't have iron to put in there, or there's a block, or there's something wrong, it's pretty smart. It has to put something into that ring. I'm making a ring. I need something with 2 pluses. It sticks zinc in. Zinc gets inserted, and zinc protoporphyrin is made, and it's absolutely worthless. Does nothing for you. And it accumulates in the cell. If it gets inserted into hemoglobin or myoglobin, you're not going to be able to hold on to oxygen. Zinc doesn't hold on to oxygen. And so I thought maybe I could just measure zinc protoporphyrin in these patients' blood. And I'll show you the data. I took healthy controls. Each dot represents a person. I looked at asthmatics. They have lung diseases, but they never get pulmonary hypertension. And I took my pulmonary hypertension patients, and I measured the zinc protoporphyrin in their blood. The levels should be under 80, and you can see the majority of patients with asthma and the healthy controls are below that number. But you can see almost all the pulmonary hypertension patients were higher. They weren't iron deficient, we checked. Now there are some controls in asthmatics at higher levels. Makes you wonder if they have some mitochondrial abnormality as well, but they don't have the disease. Nevertheless, this was a very good sign to me that this could be used to look at mitochondrial function. Now, when you don't make heme and you don't make hemoglobin, you're still making red cells. You got hemoglobin, maybe it has zinc protoporphyrin in it instead of heme, but you're making them. Usually when you make red cells, they're all the same size. They're pretty boring. They're just the same size. And we can measure the width, and the width is pretty normally distributed. If you're not making heme, and the cells aren't getting enough iron in their hemoglobin, you suddenly have a problem with red cell production. Some are very tiny, some are very big, some are normal sized. And the distribution of size of the red cells increases. And when I measured it in our pulmonary hypertension patients, they had higher zinc porphyrin. They had a distribution of red cells that were some very small and some very big. It was pretty amazing when I looked at the microscope under the slide and saw that. So I thought, okay, what could we do to test whether or not I could somehow correct the mitochondrial abnormality? Well, the mitochondria is very dependent on this coenzyme Q. And coenzyme Q is absolutely essential for this first start of the electron transport chain at both 1 and 2. You need 1 or 2 to get electrons transferred over to 3 for oxygen use. And so coenzyme Q might be a possibility for treating my patients. And it wasn't a completely new idea. In fact, there was a study ongoing when we thought of this that was for left heart failure. So patients with pulmonary hypertension have failure of the right heart, but left heart failure is not uncommon. And there was a study going on in Europe where they thought that they could improve mitochondrial energy production that might give more strength to the left heart for better vigor of pumping. And so you could maybe help improve heart failure. Now, coenzyme Q is produced in the body. The very highest concentrations are in the heart, just like the greatest number of mitochondria. I showed you 1 mitochondria for every little cell muscle pumping. And it's been used to treat rare disorders of mitochondrial function. And at the time, there were preliminary reports from this trial in heart failure. It was called Q-Symbio. There were 17 centers in 8 countries. They randomized over 400 people with severe or moderate heart failure. And they gave them 100 milligrams of coenzyme Q 3 times a day. The rest received a placebo. In 2 years' time, 25% of the placebo group had had an adverse event, cardiac event. And 17% of those placebo patients had died, in compared to only 14% of the co-Q receiving patients having an event, and only 9% of the co-Q10 patients died. It was half as many events, half as many deaths. And co-Q10 now, if you are looking, this is published, and many people with heart failure do take this drug. And they think it's because it relates to improvement in cardiomyocyte function. So I thought, let's try this in our pulmonary hypertension patients. And I decided to use the same dose. I gave them 100 milligrams 3 times a day, and I said we would do this with our patients for 3 months. I needed a comparator group, and I chose not to give placebo to the patients, but rather to take healthy people and give them coenzyme Q. Why was I doing that? I actually didn't have any way of measuring the mitochondrial function except for looking at the zinc protoporphyrin and the size of the red cells. So I thought I would look at that, and if I saw improvements in those things, then I could look at the heart function and see if heart function had improved. And as I showed you on that echocardiogram, it's easy to measure heart function with an echocardiogram. Just rub the probe on the chest and we can measure it. And so we started the study. It was extremely easy to recruit for this. Coenzyme Q has really no side effects. So the red dots will always be controls, and the blue are the pulmonary hypertension patients. And at the beginning of the study, at baseline, pulmonary hypertension patients and healthy controls had the same amount of coenzyme Q. I was actually one of the controls in this group. And as I took, and the other healthy controls took their CoQ, levels did rise. And at 12 weeks or 3 months, they were pretty high. The pulmonary hypertension patients all increased to a greater degree than the healthy controls, so that by the end of their study, their levels were more than twofold significantly higher than the healthy controls. We don't know why that is, but it's definitely different. So then we looked at the hemoglobin. We wanted to see, are they having hemoglobin production, and how much hemoglobin is packed into these cells, the iron heme, not the zinc heme. And here I'm showing you at the start of the study, 6 weeks and 12 weeks after coenzyme Q in our controls and our pulmonary hypertension patients. Now, you'll notice that at the start of the study, just as I showed you with the zinc protoporphyrin, the pulmonary hypertension patients have increased zinc protoporphyrin and they have less heme porphyrin, less iron. So their levels were significantly lower. This is looking anemic almost. But as the study progressed, their levels rose, small but significant, actually increasing grams. Picograms per cell is significant in this case, so that by the end of the study, there was no difference between controls and pulmonary hypertension for their heme content, the iron heme. And most importantly, if we looked at the red cell size variation, here's our healthy controls. Their size is the same size and it's uniform, rock solid between 12 and 13. I hope you're all that in this room. And they didn't change over the study. Our patients, I told you, had levels 15, some big, some little. The range is variable. They're significantly higher, and at 6 weeks, no real change. But by 12 weeks, the size has come down to a healthier level. 14 is actually the cutoff for normal. So we had impacted the red cell production, the heme iron, which occurs in the mitochondria. To us, this was pretty good evidence that the mitochondrial abnormality could be related to electron transport chain. So what did the heart do? Most studies of heart disease go on for 2 years. You saw the Q-Symbio study, because you're looking for outcomes like death. And thankfully, people don't die within 3 months, not that quickly. So 6 months would have been a good time period to look for outcomes of events. But we had chosen 3 months, partly because patients were doing this and hearing about it. And I will tell you, everybody wants to go on this drug who has this disease. It's amazing to me. There's websites with it. But when you look at the heart function, even in this 3-month period of time, we could see that the coenzyme Q at this dose improved heart function significantly in a paired fashion. And this kind of improvement is so important to the patients because the primary determinant of outcomes with pulmonary hypertension has to do with your right heart function and how well it pumps. So we were very pleased with this, and we're moving on, and others are trying different kinds of compounds to affect the mitochondrium. And we're starting to target different aspects of the mitochondria. You can see in some of these patients, there wasn't improvement. And it was a short period of time, but yet, we know that there will be other targets in these patients. So we're continuing to work on this disease and try to understand the mitochondrial limitations to respiration, to heme production, red cell production, and cardiomyocyte function. And what ties all those things together is the mitochondria, where the heme is produced, which makes the energy for the heart to keep pumping vigorously, and which consumes the oxygen coming from the lungs and releases the CO2 released by the lungs. So with that, I think I will end. This is a lovely picture of Cleveland on the lake, downtown. And I welcome you all to visit me. I'm here at the Cleveland Clinic, right here. This is Carnegie, Euclid, And of course, we have our Republican National Convention. It's dangerously close to the Cleveland Clinic. Yes. So we'll be here. I won't be going down there. Thank you.
The Origin Science Scholars Lectures are presented by Case Western Reserve University's Institute for the Science of Origins, with the assistance of the Siegel Lifelong Learning Program, the College of Arts and Sciences, and MediaVision. For more information on the Origin Science Scholars Program, including a full video archive, please visit the Institute's website at origins.case.edu.